Food. Food is the most vital aspect in humanity's existence. A person who doesn't have enough food to survive will not be able to develop further and will finally die. For addressing the food demands of expanding population, agricultural productivity has to be increased by 2050. It is vital to maximize efficiency and benefit from the yield in order to meet the world's food demands, while also growing farmlands. Plants generate food, which might be considered the lifeblood of our species. However, the plant may not always be able to provide the necessary food due to its external factors, and that would be the stress. Stress is a term used to describe an external influence that inhibits plant growth. It can be biotic, which produced by living things, and abiotic, which induced by non living things, like temperature, mineral toxicity, and salt stress. For today's video, we're going to focus on salt stress. To live in a saline settings, plants must adopt their internal chemistry. Carbohydrates, amino acids, and compatible solutes can assist the plant in lowering its water potential. The main disadvantage of employing this tree is that they demand a lot of energy. Thus, moderating the entry and outflow of ions might be a possible choice. The mediation of ions play an important role in lowering plants' water potential, meaning the plant should accumulate more ions in order for it to attract water and to remain turgid. The only problem with this is too much ions can be toxic to cytosolic enzymes. To minimize toxicity, ions must be stored and accumulated in the vacuoles. And because sodium chloride is the most abundant salt in salinity stress, transport proteins that store sodium in vacuoles are essential. Calcium is also an important component in alleviating salinity stress because it helps by signaling particular proteins to promote potassium and sodium selectivity. The pH levels and membrane potential are two more critical factors that influence ion homeostasis. The adjustment in pH levels is likely related to the migration of hydrogen ions in the vacuoles and in salt-tolerant plants. A combination of cytosolic alkalization and vacuole acidification is discovered. Fortunately, there is a sodium and hydrogen antiporter that can be found in the vacuoles, which is extremely useful for the plants since this is the major protein that stores and accumulates sodium in a vacuole. Membrane potential is also significant since it has the ability to transfer ions in and out of the cell. Positive ions are drawn to the negative side of the membrane, whereas the negative ions are drawn to the positive side of the membrane. Maintaining a negative potential within the cell is critical for salinity tolerance because it keeps a high concentration of potassium inside of the cell. Maintaining a balanced sodium and potassium ratio inside the cytosol, which is also important for salinity tolerance. The hydrogen pumps, or hydrogen triphosphatase, are the major proteins responsible for the shifting of pH levels and membrane potential. As salinity increases, so does the activity of these hydrogen pumps. Another protein that facilitates active transport is the SOS1 gene, also known as salt excessively sensitive gene. Operates as sodium hydrogen antiporter, meaning it transports hydrogen into the cell and sodium out of the cell, and this protein is also controlled by two additional sets of genes, which is the SOS2 and SOS3. Another protein located in the plasma membrane is the KOP1, or potassium uptake transporter, and the TRH1, or root hair gene, whose name suggests that it's involved in the development of root hairs. Both these genes function as potassium absorbers, which is essential for keeping potassium to sodium ratio. Another potassium transporter is the ATHK1, also known as Arabidopsis thaliana high affinity potassium absorber. And when its capacity to take potassium is blocked by the salt, the protein will now operate as a selective sodium transporter controlling sodium absorption. To maintain calcium homeostasis, the gene ACA, which stands for auto-inhibited calcium adenosine triphosphatase, pumps calcium from the cytosol to the extracellular space. And the ACA in the tonoplast pumps calcium from the cytosol into the vacuole. CAX1 and CAX2, which stands for vascular calcium and hydrogen antiporter, translocate calcium and other metal ions into the vacuole via the autogradient created by two hydrogen pumps in the tonoplast. Finally, AT-NHX, also known as Arabidopsis thaliana sodium and hydrogen antiporter, is in charge of sodium storage in the vacuole. Ion compartmentation in the vacuoles reduces the cytosinic damage caused by ion toxicity. The compartmentation of sodium into the vacuoles is an effective method for avoiding the harmful effects of sodium in the cytoplasm. Furthermore, sodium compartmentation into the vacuole allows plants to employ sodium chloride as an osmoticum, which maintaining an osmotic potential that forces water into the cells. Cells activate a calcium channel in response to salt stress, which raises a total cytosinic calcium. The increase in calcium triggers the SOS cascade by binding to a calcium binding protein called SOS3 and another protein called SOS2, which is a serine threonine kinase that utilizes phosphates for energy. SOS2 will then be activated SOS1, a sodium and hydrogen antiporter, causing excessive sodium to be extruded from the cytosol. SOS2 kinase also interacts with other tonoplast proteins such as CAX1, NHX, and hydrogen adenosine triphosphatase, indicating that SOS2 plays a central role in regulating transport proteins. In conclusion, maintain a healthy sodium and potassium ratio, calcium in the cytosol should be increased, increase the activity of hydrogen pumps, sodium in the vacuole is partitioned, the SOS cascade is activated. These are the methods by which plants activate their stress tolerance. Plant-specific genes may aid in the development of tolerance in plants. That is why scientists are focusing their efforts on it in order to assist humanity in surviving on plants for nourishment. These suggestions may all aid in identifying which genes can be used for scientific purposes, such as on expressions and the likes. Without plants, animals, or should I say humans, would have no oxygen to breathe and would die. Without plants, there would be no food to eat. According to the Chinese proverb, he who plants a garden plants happiness. Thank you. I'm RJ Flores, and I am your plant to the rescue.